Hello students, in last video we have covered interphase of the cell cycle, and in that we have seen three phases G1, S and G2, and we have also discussed an extra phase which is not part of interphase, which is known as G0 phase. Okay, so G0 is not part of interphase. We have discussed all these phases in last video. And today we are going to start the next phase of the interphase, that is M phase. Okay, we all know that M phase is very short phase of the cell cycle, and during M phase the cell will divide. During the M phase, cell divides and either two new cells are produced or four new daughter cells are produced from the parent cell. And we will talk about the M phase today. Okay, so M phase is the shortest phase of the cell cycle, and during this phase the cell divides into two daughter cells or either four daughter cells. Okay, so let's learn about that. And in the previous video we have seen that M phase can occur via mitotic method or it can occur via meiotic method. Okay, M phase can take place via mitotic method or via meiotic method. It can occur via two methods, mitotic method and meiotic method. When the M phase occurs via mitotic method, it occurs generally in diploid cell or haploid cell. Okay, it occurs in both diploid as well as haploid cell. When the M phase occurs via mitotic method, this type of method is seen in both diploid cell as well as haploid cell. And when the M phase occurs via meiotic method, it occurs only in the diploid cell. Now you will get confused about these terms. What is diploid cell and what is haploid cell? Okay, so diploid cell is that cell which is having two sets of chromosome. Diploid cell is that cell which is having two sets of the chromosome. We receive one set of chromosome from our father and one set of chromosome from our mother. So in this manner we have two sets of chromosome. Okay, so we are diploid in nature. We can say that we are diploid in nature. Okay, so a diploid organism or a diploid cell has two sets of chromosome. One set of chromosome is received from father and one set of chromosome is received from mother. Whereas haploid cell has only one set of chromosome. Haploid cell has only one set of chromosome. Say for example, this is a diploid cell is there, so it will be having 2n number of chromosome. 2n number of chromosome means it will be having n plus n set of chromosome. One set of chromosome is known as n and another set of chromosome is known as n. So both together are known as 2n. So it is known as diploid organism, okay, or diploid cell. And this haploid cell will be having only one set of chromosome which is represented as n. Okay, so diploid organism can be called as 2n organism and haploid organism can be called as n organism, or we can say diploid cell is known as 2n cell and haploid cell is also known as n cell. Okay, so mitotic method can occur in diploid cell as well as haploid cell, whereas meiotic method can occur only in diploid cell. Meiotic method will occur only in diploid cell. Okay, meiotic method will occur only in diploid cell. This is the difference. This is the key difference between the mitotic method and the meiotic method. Okay, when M phase takes place, it can occur via two method, either via mitotic method or via meiotic method. When the M phase occurs via mitotic method, it can occur in both diploid cell as well as haploid cell. Whereas when the M phase occurs via meiotic method, it can occur in the diploid cell only. Now what is diploid and haploid? A diploid cell is that cell which is having two sets of chromosome, whereas a haploid cell is that cell which is having only one set of chromosome. Diploid cell can be represented as 2n. It is having 2n number of chromosome, whereas haploid cell has only n number of chromosome. Okay, here also diploid cell has 2n number of chromosome. So this is the key difference between mitotic method and meiotic method. Here also I have explained the difference between mitosis and meiosis. When the cell divides via mitotic method, it will give rise to two daughter cells. It will give rise to two daughter cells. When the parent cell divides via mitotic method, it gives rise to two daughter cells. But when a parent cell divides by a meiotic method, it will give rise to 4 daughter cells. Okay, when the parent cell divides by a meiotic method, it will give rise to 4 daughter cells. Okay, this is the second difference between the mitotic method and meiotic method. First difference is mitosis occurs in both diploid and haploid cell, whereas meiosis occurs only in the diploid cell. Second difference is in mitosis 2 daughter cells are produced from the parent cell, whereas in meiosis 4 daughter cells are produced from the parent cell. This is the second difference. And the third difference that I am going to tell you is that the daughter cell which are produced from the parent cell in mitosis, the daughter cell which are produced from the parent cell in mitosis are identical to the parent cell. They are identical to the parent cell. Okay, they are identical. These cells are identical to the parent cell, or we can say these cells are copy of the parent cell. These cells are copy of the parent cell. When the cell divides by a mitotic method, the daughter cells are identical to the parent cell, or we can say that daughter cells are copy of the parent cell. And when the cell divides by a meiotic method, the daughter cells are different from the parent cell. Daughter cells are different. Daughter cells are different from parent. These cells are not similar and they are not same as parents. Okay, they are not same as parents. No, not similar. Similar term we will not use. We will use the term different. They are different from their parent. Okay, or we will say that they are not identical to their parent. They are not identical to the parent cell. Okay, so when the cell divides by a mitotic method, the resulting daughter cells will be copy or identical to the parent cell. But when the parent cell divides by a meiotic method, the resulting 4 daughter cells are different from their parent. They are not identical to the parent. So this is the key difference between mitotic method and meiotic method. Okay, we are discussing the M phase of the cell cycle, and we said that M phase is the shortest phase of the cell cycle, and during this phase the cell divides. And here 2 methods can be applied. One is mitotic method and one is meiotic method. When the cell divides by a mitotic method, it is found only in diploid cell. It is found in both diploid cell and haploid cell. But when the cell divides by a meiotic method, it is found only in the diploid cell. Okay, and what is the meaning of diploid and haploid? The diploid means that the cell is having 2 sets of the chromosomes which are received from mother. One set of chromosome is received from mother and another set of chromosome is received from father. Okay, whereas the haploid cell will have only 1 set of chromosome. Okay, so in mitosis can occur both in diploid cell as well as haploid cell, whereas meiosis can occur only in the diploid cell. And the second difference is, after mitosis 2 daughter cells are produced, and after meiosis 4 daughter cells are produced. Then third difference is that after mitosis the resulting 2 daughter cells are exact copy or they are identical to the parent cell, whereas in the meiosis the resulting 4 cells are different from the parent cell. Okay, chalo. Now we will learn mitosis in detail. Okay, so process of mitosis has 2 steps. One is karyokinesis and second is cytokinesis. We all know about karyokinesis. Karyokinesis is nuclear division and cytokinesis is cytoplasmic division. Okay, when the M phase occurs via mitotic method, mitosis has 2 phases, karyokinesis and cytokinesis. Karyokinesis is known as nuclear division and cytokinesis is known as cytoplasmic division. Now this nuclear division is again divided into 4 subphases. Nuclear division has also 4 steps or 4 subphases. They are prophase, metaphase, anaphase, telophase. Prophase, metaphase, anaphase and telophase. These are the 4 subphases of the karyokinesis. Okay, or we can say that karyokinesis occurs in 4 steps. Karyokinesis occurs in the 4 steps. They are prophase, metaphase, anaphase and telophase. Prophase, metaphase, anaphase and telophase. Okay, so what is the summary that summary is that during the cell cycle, the cell will pass through G1, S and G2 phase. Okay, cell will pass through G1, S and G2 phase, and then it will enter into the M phase. And when M phase occurs via mitotic method, it has two steps in it. One will be karyokinesis and second will be cytokinesis. Okay, and karyokinesis has four sub steps or four sub phases: prophase, metaphase, anaphase and telophase. After the completion of telophase, cytokinesis will occur, and after cytokinesis, two daughter cells will be produced. After cytokinesis, two daughter cells will be formed. Okay, so we are going to learn this mitosis. We said that mitosis has two sub phases: karyokinesis and cytokinesis. Karyokinesis has four sub phases: prophase, metaphase, anaphase and telophase. After completion of telophase, cytokinesis will occur, and after cytokinesis, two daughter cells will be produced. Today we are not going to cover this topic. Okay, today we are not going to learn meiosis. We are learning mitosis. Okay, so we'll learn mitosis in detail. So students, as we said earlier, that we are going to learn M phase, and in M phase we are covering mitosis today. And in mitosis also we are going to learn karyokinesis first, because karyokinesis is the first step of mitosis. Okay, so we will learn mitosis first, and then we will learn meiosis. Okay, so we will learn mitosis first, and in mitosis also we are going to cover karyokinesis first. But before that, let us revise what has happened in the interphase. Okay, so in the interphase, three subphases are there: G1, S and G2. Three subphases are there in the interphase. Okay, in the G1 phase, cell was like this. It was having intact nuclear membrane. It was having a centriole. Cell was having chromatin fiber. Okay, in the interphase, chromatin fibers are there. Okay, and cell has mitochondria also, cell has nucleolus also, and cell has chloroplast also. See, I am drawing mitochondria and chloroplast and centriole also. See, in animal cells, centrioles are there, but chloroplasts are not there. Okay, and in plant cells, chloroplasts are there, but centrioles are not there. So don't get confused that whether this is a plant cell or animal cell. This is a cell only. Okay, we are not differentiating between plant cell and animal cell now. Okay, we are just learning cell division. So you can say that this can be a plant cell also, it can be a animal cell also. Don't get confused in that. So a centriole is there, mitochondria is there, chloroplast is there, intact nuclear membrane is present, nucleolus is there, and chromatin fibers are there in the G1 phase. Okay, now when this cell enters into the S phase, remember the events of the S phase. The centrioles get duplicated. Here one centriole was there, here two centrioles will be there. Okay, then here mitochondria and chloroplast will be there in one number only. See, mitochondria and chloroplast will be one in number. One centriole was there, now it will have two centrioles. Then here the chromatin was there, and here newly synthesized chromatin is present. Okay, initially it was having this much chromatin. Now this cell will be having this much plus this much extra chromatin. Why? Because new chromatin has been synthesized, new DNA has been synthesized in the S phase. That's why the amount of the chromatin will get doubled. So we are saying that in the G1 phase, cell was having one centriole and some amount of chromatin. But in the S phase, the centrioles will be duplicated and double amount of chromatin will be there because of the DNA synthesis. Now the cell will have double amount of chromatin, and it has intact nuclear membrane, and it has nucleolus also, it has mitochondria and chloroplast in the same number as were present in the G1 phase. Okay, the number of the mitochondria and chloroplast has not yet increased. Okay, so their number is same which were present in the G1 phase. Now when this cell enters into the G2 phase, again same number of the duplicated centrioles will be there. Okay, 2 centrioles are there. Same amount of the chromatin is also there. Duplicated chromatin is there. Okay, duplicated chromatin is also present. Intact nuclear membrane, nucleolus is also there. But what is the difference? Mitochondria has got doubled, and chloroplast has got doubled. Okay, it has been duplicated. Mitochondria and chloroplast number has been increased. Okay, so in the S phase, mitochondria and chloroplast were 1. But in the G2 phase, there will be 2 mitochondria and 2 chloroplast, or double the number of mitochondria and double the number of chloroplast will be there. See, remember the events of G2 phase. In the G2 phase, chloroplast and mitochondria are increased in number because of the fission. Okay, so this is G1 phase, this is S phase, and this is G2 phase. Now, after the completion of G2 phase, the cell will enter into the M phase. In the M phase, it will enter into the mitosis. In the mitosis, it will enter into karyokinesis, and in the karyokinesis, it will enter into the prophase. It will enter into which phase? Prophase. And this prophase is also again divided into 2 subphases. Prophase is also divided into 2 subphases. This is early prophase and this is late prophase. This is early prophase and this is late prophase. We are going to discuss karyokinesis. We are discussing karyokinesis, and in that we said that cell has entered in the prophase, and the prophase has also 2 subphases, early prophase and late prophase. This is early prophase and this is late prophase. Now see here what has happened. That in early prophase, what is the difference between this cell and this cell? What is the difference between this cell and this cell? Here the chromatin fiber has started constricting, okay, or constriction of the chromatin fiber has begun. Okay, in this phase the constriction of the chromatin fiber has begun, and now it is taking shape of chromosome. It is taking shape of chromosome. In the G2 phase, the chromatin were present in thread-like form. In the G2 phase, chromatin were present in the thread-like form. But here, the chromatin condensation has begun and it has taken the shape of chromosome. Here, the chromosome has been produced, or chromosome formation is going on. Okay. In the same manner, the centrioles has started to move towards the pole. Centriole has started to move towards the pole, and nuclear membrane has also begun to be fragmented. Okay, nuclear membrane has also is also being destroyed over here. Okay, since nuclear membrane is also becoming fragmented, or it is also becoming destroyed. Okay, here the intact nuclear membrane was there, but here the fragmentation of the nuclear membrane has started. Fragmentation of the nuclear membrane has started. So what are the key events of early prophase? Early prophase. What we will say in the early prophase? We will say that chromatin condensation has begun. Chromatin condensation begins in the early prophase. Then we will say that nuclear membrane disintegration starts in the nuclear membrane disintegration begins in the early prophase. Okay, then we will say that centrioles started to move towards poles. Centrioles have begun to move towards poles. Okay, this is all about early prophase. This is all about early prophase. Okay, in the early prophase, what are the key events, or what are the key features of the early prophase? In the early prophase, chromatin condensation begins, nuclear membrane disintegration begins, and centrioles start to move towards the poles. These are the key events or key features of the early prophase. Okay, and now when the chromatin condensation has begun, the chromatin will take shape of the chromosome. In the G2 phase, chromosomes were not visible, but in the early prophase, thin chromosomes are beginning to be visible. Okay, or they are becoming visible. Okay, this thin thread-like chromosomes are becoming visible now because of the chromatin condensation. Okay, sure. Now let's talk about late prophase. This is late prophase. Okay, this is early prophase. This is late prophase. What happens in the late prophase? The centrioles are still moving towards the pole. Centrioles are still moving towards the pole. They are moving towards the pole. Chromatin condensation is still going on. Chromatin condensation is still going on. Okay, or it is increasing further. Then nucleolus has been disappeared. Nucleolus is not there. Here nucleolus was present, but here nucleolus is absent. So nucleolus is not there. Okay. Then I have not drawn anything apart from this. Okay, but Golgi body and endoplasmic reticulum has been lost or they are destroyed. Golgi body and endoplasmic reticulum are disintegrated or they are removed. Okay, so what are the key events of late prophase? In the late prophase, centrioles are still moving towards the pole. Nucleolus is absent. Nuclear membrane disintegration has increased or it has reached to next level. Okay, here nuclear membrane disintegration has begun. It was just begun. Okay, or it has just begun, but here it has increased further. Okay, here it has reached to a next level. Okay, then we will say that Golgi body and endoplasmic reticulum has been removed. Okay, or they are lost. Okay, so what are the key features of late prophase? Late prophase. Chromatin condensation continues. Chromatin condensation continues. Okay, then we will say nuclear membrane disintegrates further. It disintegrates further. Then we will say nucleolus is lost or nucleolus is removed. Then we will say that endoplasmic reticulum and Golgi body is removed. Endoplasmic reticulum and Golgi body is removed. Clear, child? So this is all about late prophase. Okay, in the late prophase, what are the key events? Chromatin condensation continues, nuclear membrane disintegrates further, nucleolus is lost, endoplasmic reticulum and Golgi body is also removed. So these are the key features of the late prophase. Okay, we are learning karyokinesis, and in karyokinesis there are 4 subphases: prophase, metaphase, anaphase and telophase. In the prophase also there are 2 subphases: early prophase and late prophase. The key events of early prophase are chromatin condensation begins. Okay, nuclear membrane disintegration begins. Centrioles start to move towards opposite pole. These are the events of the early prophase. Then the late prophase will take place. In the late prophase, chromatin condensation continues, nuclear membrane disintegrates further, and nucleolus is lost, and endoplasmic reticulum and Golgi body is removed. Okay, so we have learned early prophase and late prophase in the karyokinesis topic. Okay, I will keep up to this only. In the next video we will learn about metaphase, then we will learn about anaphase, and then we will learn telophase. Okay, so we are keeping up to this only. Please watch the video carefully 2-3 times and then submit your homework after watching the video and reading the textbook. Okay, best of luck.